Project Mercury is one of the research projects about to board SpaceX, headed for the International Space Station. This research project is made possible by Space Florida and NanoRacks, and it involves 3 organizations: UC Davis and the ISSRC, SciStarter, an online citizen science community, and Science Cheerleader, an organization of more than 250 current and former NFL and NBA cheerleaders who are also scientists and engineers. The research has 3 components. The one we're extremely excited about today, for obvious reasons, will be heading via SpaceX to the International Space Station. And these are 4 carefully selected microbes. The microbes will be compared for their growth rates on the International Space Station, which, as we know, has a near zero-G environment. We'll look at their counterparts left on Earth at the UC Davis lab. We'll compare their growth rates to each other, and we'll also ask the astronauts to swab the inside of the space station. So they'll be sending us samples of microbes, germs, that live on the International Space Station. So that's 2 components of this research project. The third, which is also very exciting, involves thousands and thousands of people from all over the country who have been collecting microbes from their own shoes and cell phones. All of those samples get sent to Argonne National Lab, where they're sequenced and fed into what's called an Earth microbiome map. All of this is available and open to the public. You can go to spacemicrobes.org to see what we're finding. You can go to spacemicrobes.org as well to understand the growth rate comparisons that are happening on the space station. As soon as we find out, you'll find out. You'll also learn there what kind of microbes the astronauts did collect from the space station. And I would like to add one thing. The samples that are sent to the International Space Station are safe. They're common bacteria, and they were collected from the most fascinating places, including Liberty Bell, Sue the T-Rex, the Philadelphia Phillies Stadium, the San Francisco 49ers Stadium. So in carrying on with that sports theme, when we have this race in space, it will be positioned as a microbial Super Bowl in space, and we're very anxious to see which team wins.
The thing that I think excites me the most about this experiment, to give you a sense of how I feel when I see one of my experiments going into space, it's like nothing I've ever experienced before. When you see that craft leave the Earth's surface and go, so far above our atmosphere with the experiment that you worked very hard on for the last several years, and that you touched, and then you anticipate all the exciting science data that you're going to get back from this experiment involves using fruit flies, or Drosophila melanogaster. And we are going to look at cardiovascular function. So we're actually going to look at how the heart functions in the spaceflight environment.
And liftoff of the Falcon 9 rocket and Dragon. SpaceX 3 is underway.
The experiment we have going up to the space station, the NanoRacks experiment, is designed to study the very earliest stages of planet formation. In particular, we're going to be looking at very gentle collisions between small particles to understand how the size, density, and surface properties of those particles affect the outcome of the collision.
My name is Akram Amin. I'm an engineer at the German Aerospace Center. I'm also a graduate student at the Technical University in TUM, Technical University in Munich. My mission is called EGAHEP, or Egypt Against Hepatitis C. We are planning to send purified proteins of the hepatitis B virus in order to be crystallized in a microgravity environment, and then after it returns from the ISS, we can try to gain the crystal structure of it and try to understand its behavior and how it replicates, and after that we can use this information to target the replication process and stop it.
Energy is looking at plant-microbe interactions. We're going to take a clover-like plant and a bacteria that infects its roots, and instead of forming the disease, forms this beneficial relationship. It does some really neat stuff. It takes nitrogen that's in the atmosphere that we're going to use. That bacteria convert that to a fertilizer, and then in exchange, the bacteria asks that they take some carbon dioxide, turn that to sugars, and that supports the bacteria.
And we are Team MicroGRX. And our payload is a biochemical experiment. It will be the first solution-based experiment to be tested on the plate reader that's up on the space station. And so there's really three purposes to our payload. So first is to test the technology. Two is to validate this plate reader, which really hasn't been used. It certainly. And then so the third is to actually do a biochemical experiment. And this experiment is measuring a vitamin found in milk binding to an antibody.
2, 1, 0.
So we call the experiment SABLE, which stands for Self-Assembly in Biology and the Origin of Life. We're making a small little chemistry lab in a hole that's 4 inches by 4 inches by 6 inches. And within that we have 9 vials that we grow long thin protein fibers in. And they self-assemble in a vial essentially. And we do that in the laboratory. We've been doing it for a number of years. They go through a series of phases where they units that grow long fibers. The fibers then begin to settle in the solution. They sort of lock over each other and get into more complex forms. We are going to do it in weightlessness.